Okay, Prem Tej, right?
Yes, sir.
Okay, Prem, are you okay?
Yes, sir. I'm completely fine.
Okay, ready for mock interview?
Yes, sir.
Okay, Prem, introduce yourself.
Sir, my name is Prem Tej, and I come from Kolar district of Karnataka state. I have completed my BSc from Bangalore University in 2019, and with chemistry, botany, and biotechnology as the core subjects. And I have completed my post graduation in biotechnology from University of Mysore in 2021. Since then, I was preparing for some competitive exams, and I haven't been to any job yet. And my hobbies are so watching cricket and playing cricket, and pencil sketching, etc. So this is my brief.
Okay, so Prem, your hobbies is watching cricket, right?
Yes, sir.
So
I'm
currently,
playing too.
IPL is going on. How many teams are there in IPL?
Sir, currently Rajasthan Royals stands at the top, and our own home team RCB ends up last in the point table.
Total number of a team in IPL 2024.
Sir, total 10.
10. Okay,
Yes.
and who is your favorite player?
Sir, Virat Kohli, sir, Mr. Virat Kohli.
Virat Kohli. Why? What
Sir,
you what you
it's
most like in Virat Kohli?
Sir, his intent in playing, when it comes to his uh, game, I usually like his class in batting, and I like the way he approaches, and I like the way chases the score. And when it comes to the personality, he is one among many sportsmen, or few sportsmen we can say, who are dedicated very much to their sport, who contribute a lot to the sport, and bring that dignified to the sport. And when it actually the hunger that he has to win games for his nation, and alongside he even makes out a good gestures being on field, like we can say he was stopping the crowd from booing for Steve Smith, or booing against Navin Hulak, or booing against Hardik Pandya recently. These kind of gestures and the sportsmanship what he uplifts every time in every game is what actually astonishes me. And along with this, he gives his time for his family. He balances the work and both family, the work life balance that he creates or he shows to the society is something which has to be appreciated, sir.
Okay, Prem, you are a huge fan of Virat Kohli. Probably
Sir.
you explain all the positive things in Virat Kohli, like as his gesture, his dedication, his fitness, and contribution to the sports, and his hungerness about the winning of match for the country. So all those things, very well done. Prem, you are going for a
Yes, sir.
CPTM patent officer's interview, right?
Yes,
So
sir.
why you think that you are a good candidate for the CPTM patent officer?
Uh, Sir, I can't say that I'm better or best than the other 249 members who are competing along with me. But sir, there are few inherent qualities in me which I could say, and that would match the requirements of the CGPDTM examiner of patents post. So speaking about that, uh, I'm usually a team person. I love to be in a team, and I love to contribute something to my team when we are assigned a work, especially. So that could help me in interacting with my fellow colleagues if selected. And sir, the kind of patience that I have, because if someone approaches me for applying for a patent, and if something goes wrong or something like that, I'm patient enough to listen to them and take the necessary steps. And sir, we can say I do have some procrastination kind of thing, but when some work is assigned to me, I'm really dedicated to it. And the post of CGPDTM, it actually requires one to be attentive, one to be focused when analyzing the patent application. So when it comes to that, I have this analysis skills. I can analyze the data. I can interpret the data. So which quietly matches the requirements for the CGPTTM post.
Okay, well done, Prem. As everybody has their own limitations and drawbacks and negative points, etc. Right? So,
Yes,
if I
sir.
asked you, what is the negative and drawbacks in you?
Uh, so basically, I'm an introvert person. So it usually takes some time, not much time, a little time to get adjusted or speak openly or freely with the person whom I met. So, but that could be overcome, sir, because every time I visit a new institute for my studies, obviously the persons are new, but initially I might face a little trouble in interacting with them, but ultimately I would get back. I'll manage that. And the second thing is, as you said that I had a little bit of knowledge about cricket, and cricket itself is a little distraction for me because if while I'm studying or something, and if someone calls me for cricket, I'll just put that aside and I'll go back. And this I have to reduce. But if the work is really very serious, then I'm committed to it. Uh, so that's it, I think.
Okay, Prem, you did your MSc from the Mysore University, right?
Yes, sir.
So can you tell me the positiveness of that Mysore University? How Mysore University is good for you and for other?
Sir, I would state that University of Mysore is the top university among universities of Karnataka, which is why I got enrolled into it. And then, sir, the professors over there are well qualified, are senior most, and they have a wide variety of knowledge regarding their specific subjects. And if we talk about the campus, it's too good. And if we talk about my department, especially, there are very good works going on, good project was going on, and my department is especially it's famous for the work that is going on downy mildew disease of pearl millet. So it has been continuously researching on this disease from past I can say so 10-15 years. They have been funded, and I'm glad to be a part of that university, sir.
Good, well done. Prem, you are from the Karnataka itself, right?
Yes, sir.
So can you elaborate five key points which are very very attractive for the peoples who are going to travel in Karnataka?
Sure, sir. So the first thing is let's talk about tourism, sir.
Yeah.
So when it comes to tourism sectors, we got Western Ghats, we got Chikmagaluru and Coorg, uh, which is most visited place by everyone to visit in the winters. And when it comes to the coastal areas, we got beautiful beaches like Maravante Beach, Gokarna Beach, Honavara Beach, etc. And it when it comes to historically, if we start talking, we have got ancient temples like Hampi, Pattadakallu, Badami, Aihole, these kind of historical places we have. And when we talk about if someone is fascinated about technologies or software or something, obviously we got Bangalore, which is an IT hub, which is an electronic city, garden city. So one can get their like if someone is willing to work and under a good ambience or a good atmosphere, obviously Bangalore is always cool. The climate over Bangalore is much appreciated by everyone because of its neutrality that it maintains. So yes, and rest two things. Yeah, obviously, if you want to watch cricket, you can visit Chinnaswamy Stadium in Karnataka. It gives you a great experience. And sir, when it comes to educations, we Karnataka we have IISc, and we have got an institute for plant biotechnology separately, uh, and CSRIT for Silk Research Institute. We have got CFTRI, Central Food Technology Research Institute, for educational purposes. Yes, sir.
Okay. Prem, can you tell me the some research institutes funded by the CSIR, ICMR, or ICR situated in the Karnataka itself?
Uh, Sir, one is CSRIT, I think, Central Silk Research Institute.
Okay.
That and sir, IISc, Indian Institute of Science, and obviously CFTRI, sir, Central Food Technology and Research Institute.
Okay.
Sir, and I'm not recollecting. further, sir.
Okay, Prem, what is your area of domain and favorite topic and subject?
Sir, I would like to give my first priority to plant biotechnology and then to bioprocess engineering, sir.
Okay, well done. Plant biotechnology, right?
Yes, sir.
So, what is bioremediation?
Sir, we can say it is a process which is used to eradicate or lessen the toxicity or pollutants present in either soil, water, or in the environment by using microbes or other biological agents as a medium, sir.
Okay, Prem, suppose you are dealing with some microbes and you don't know anything else about that microbes, right? And you are dealing in the laboratory. How you can say that these microbes are not pathogenic for us? On the what basis you can say that these bacteria are these microbes or bacteria or funguses are not pathogenic for us? What will be the criteria for identify the pathogenic or non-pathogenic?
Sir, without a minimum knowledge of the microorganisms, we cannot assume or we cannot conclude that this microorganism is pathogenic or not. We have to have at least a bare minimum knowledge about the microbes. And sir, we can say like in our university we were just allowed to handle those microorganisms, and we were allowed to inoculate only those microorganisms which are less harmful. And if we say this, we were never exposed to any microorganisms that are harmful. So currently I don't have any idea to distinguish microorganisms on the basis of toxicity or not.
Okay, for general purpose, for growing of a bacterial cultures in microbial laboratory or any laboratory, we generally use shaker, right? BOD shaker.
Yes, sir.
So
Yes, sir.
what is the temperature of that shaker
uh,
for the optimum growth of bacteria?
sir, sir, it do varies for different microorganisms, sir, because few
You
might
can
be
say what
thermophiles.
is the range?
Sir, 28 degrees Celsius to 32
28
is
degrees
what I
Celsius.
to 32.
Okay, Prem, have you ever grown any bacteria at the 37 degrees Celsius temperature?
Sir, I don't remember, sir.
No need to say. Okay,
I
okay,
don't remember, sir. Yes.
okay, no issue. So, Prem, can you suggest some plant vector?
Plant vector.
As you are interested in the plant biotechnology, so
Yes, sir.
yeah,
Plant vector, plant-based vector. I'm not able to.
yeah, a plant-based vector like just say suppose like we have a bacterial vector, animal vector, plant vector, right? Suppose like we have
Okay.
a bacterial vector that is a plasmid, cosmid, phasmid,
Yes, sir.
etc.
Yes, sir. Yes, sir.
Viral vector, viral-based vector. Yeah, you can say that plant-based vector.
Plant-based vector. Sir, sorry, sir. I'm not able to
Okay,
recollect.
no issue. Prem, have you heard about the shuttle vector?
Yes, sir. I do heard.
Yeah.
I do heard about it.
What is shuttle vector?
Um, Sorry, sir. I haven't gone through it yet.
Okay, no issue. In genetic engineering, that means basically RD
Sir.
technology. RD technology,
Okay, sir.
there is a key enzyme known as the restriction enzyme, right?
Yes, sir.
Can
Yes,
you
sir.
can you elaborate the fundamental property of a restriction enzyme?
Sir, a restriction enzyme, uh, like we can consider it as two type, like type 1 restriction endonuclease and type 2. Sir, when it comes to type 1 restriction endonuclease, it cuts the it cuts the gene 1,000 base pairs away from the recognition site. And when we talk about type 2 restriction endonuclease, it cuts exactly at the recognition site. And type 1 restriction endonuclease is non-specific, while type 2 restriction endonuclease is specific. So,
Okay, that what is the property? What are the properties of restriction enzyme? It was the class about the type of restriction enzyme, but I am
it
asking
is to
about.
cut it is to cut the gene, and which could help us in inserting the gene of our interest. It is to cut.
Suppose restriction enzymes are only present in the bacteria, right? Okay, there is no report to found in the plant or animal or other some eukaryotic cells, but only exists only in the prokaryotic cells. So suppose we
Okay.
have a DNA from the plant cells, will it be able to cut that plant DNA sequence from the plant cells?
Sir, actually, the restriction endonuclease actually requires a certain sequence where it can cut. So it's not about the type of the gene that it comes from. It needs certain set of sequence of the base pairs, the only where which it could go and cut. So I assume that even the bacterial restriction endonuclease can cut the plant genome.
Why it is known as the restriction enzyme, not simply endonucleases? The restriction enzyme is a type of
Well,
endonuclease, right? So
yes,
yeah,
yes, sir. I'll get back to it, sir.
okay, no issue. Have you heard about the TI plasmid?
Yes, sir.
TI bacteria, TI plasmid. What is
Yes, sir.
TI vector or plasmid?
Sir, it is a tumor-inducing plasmid, which, when inserted into the plant, it proliferates the cell division like enormously and starts forming uh, nodule kind of a structure in the plant roots.
Okay, what do you mean by the site-directed mutagenesis?
Sir, site-directed mutagenesis is something which we could uh, it is something that has been done to cause a mutation, but since it is spite specific, with that we can assist that it has to the mutation the sorry, the mutation has to happen at a particular site of our interest, so that if defective gene is present in especially one particular site, it's not like the site specific mutagenesis will go and alter the base sequence at that very site itself.
Okay, well done. Prem, can you suggest some techniques used in the plant biotechnology for the purpose of gene cloning, for the purpose of gene therapy, etc., whatever?
So for gene cloning,
Yeah,
I mean
regarding
transgenics.
regarding the plant biotechnology, can you suggest some techniques?
Yes, sir, we can say organogenesis, embryogenesis, and uh, like and six and androgenesis, and we can say development of plantibodies, and we can say protoplast culture, developing and somaclonal variations, and
What do you mean by the soma clonal variation?
sir, since soma clones are those cells which, I mean, somatic cells are those cells which are away from, which are not the part of reproductive thing. So the almost all the cells remain same in either in the genetic content or the characteristic wise. But in soma clonal variation, due to continuous regeneration of the plant species again and again from the cells, a slight variation in the genetic content might happen due to either due to mutations or some other factors. So even though the plants that are developed are based upon the somatic embryogenesis, since we could find some variations because of the mutation, we call those variants which are, I mean, we call those offsprings as variants. And since they are developed by somatic embryogenesis, we call it somaclonal variations.
Well done. Uh, Prem, in laboratory, generally for the plant tissue culture, we uses
Yes,
certain kind of a media. Can you
yes,
suggest
sir.
and can you just give me the example of a, bacteria based on the plant culturing or plant tissue culturing?
So you're asking me to mention the media, sir.
Yeah, culture media used
Oh,
for the
okay,
plant
sir.
tissue culture. Yeah.
Okay, sir. So one is MS media, that is Murashige and Skoog media, and another one is White's medium, and another one is Niche medium, and we can use B5 medium, and we can use blood agar medium for that. Yes, these are few examples.
Okay, Prem, tell me. there are certain approaches to improve the quality of a plant in the terms of their resistivity for the certain heavy metal toxicity to and resistivity for the abiotic cases like salinity, drought, etc., and
Yes,
also
yes,
improve the quality of a food quality or food quantity.
yes,
So
sir.
suppose you have a region from the land is suffering from the salinity, right?
Okay, sir.
And plants are
Okay, sir.
unable to grow properly and develop their plant height and grow, etc. So,
Yes, sir.
what will be the approaches to mitigate that salinity by using RD technology? How
Sir.
you can approach to mitigate that salinity?
Yes, sir. So for that purpose, sir, we have to depend upon the what is that called, sir? Stress tolerant crops, because salinity is a kind of stress that has been loaded on the plant. So for that, we have to insert uh, a gene of a gene which could give the plant the tolerance against that salinity. And currently I can't recollect the plants which are grown for this purpose, that is against salinity. But yes, sir, stress tolerant crops will be the best possible uh, solutions for to grow in the conditions where there is high salinity.
Okay, Prem, nowadays there are certain reported bacteria and fungus who have ability to mitigate the salinity, drought, heavy metal toxicity, and they are very, very efficient, and they are suggested and reported in the past nearly one decade review literatures. They might be the substitute for the chemical as well as biotechnological improvement in the crop field. So, have you heard about the bacteria like as plant growth promoting rhizobacteria, PGPR, or plant
I do
phosphorus?
heard about PGPR, sir, but I haven't heard about the bacteria which you have said for this purpose, sir.
Okay,
PGPR, I have heard.
no issue. Uh, Prem, have you heard about the ROS, reactive
Reactive
oxygen?
oxygen species.
Yeah.
Yes, sir.
What is the concept of ROS and how it is formed and how it is beneficial or harmful for the plant?
Sir, I have to go through it, sir. I'm sorry.
Okay, right now you are not prepared for that.
No, sir.
Okay, so in plant biotechnology, there are certain limitations. Like as we know, the gene cloning or genetically modified organisms are very good for us, but still there are some restrictions, and there are no freedom to use all the genetically modified plants for the cultivation purpose as well as for the consumption of human beings, right?
Yes, sir.
So
Yes, sir.
to create a genetically modified organism, what will be the basic steps? Suppose you are trying to to develop a genetically modified plant, so
Yes,
what
sir.
will be the basic steps?
Sir, first of all, we have to identify what the plant lacks, or what is that character that you have to bring out in the plant that could be commercially exploited, first thing, and which could be beneficial in nutrition value or health benefits for the person. So keeping these as and keeping the and there are few genetically modified crops against herbs, pests, insects, and all. So keeping this in view, we develop transgenics. For example, if we keep pests as the main motive, we develop BT cotton, BT corn against that. If we keep nutrition as in our mind, we develop iron fortified rice, which has a high beta carotene content and vitamin B12 content in that, and iron content in that. And if we keep health benefits, and I cannot recollect the plants that have grown. We can say beta carotene enriched carrots for health benefits, and so this would be the main criteria when we develop transgenic crops, or genetically modified crops.
Okay,
And
Prem,
if
as you are a plant biotechnology students, and suppose if I use a statement like genetically modified plants
Yes, sir.
are good or bad for us, so can you justify your statement how it is good or bad for us, genetically modified or plants, yeah, organisms?
Yes, sir. Sir, the genetically modified crops and organisms have both its advantages and disadvantages, both beneficiaries and drawbacks also. If we talk about beneficiaries, sir, as I mentioned earlier, bringing out the high nutrition content and health benefits, or bringing and developing the resistant against pests and insects would be the uh, plus points of genetic modified crops. But when we talk about negatives, sir, uh, so the one thing is consumption of these crops. Like if for example, if we develop a BT toxin, I mean, sorry, BT cotton or BT corn, if we start consuming BT brinjal and BT corn, uh, there is a chance of bioaccumulation of those toxic substances in our body. And the second thing is, not everyone would be able to tolerate the GMO crops, because few might be allergic, sir. Few might be allergic for genetically modified crops. So that would be another drawback. And second thing is, sir, still the technology has to get improved to completely eradicate the side effects that has been caused by the GMO. So since still the research is going on, we can't assume that genetically modified crops are completely safe for either for the nature or human consumption. So,
Okay, Prem, one last question. What is gene
yes,
knockout?
sir, I've gone through, but I don't recollect it, sir. I have to go through it.
Okay, well done, Prem. Thank you for your nice interactions and your responses. Now the session is open for you. If you have any query, doubt, in questions or suggestions regarding your real mock interview, so you can talk to me and you can ask me and feel
Yes, sir.
relaxed and be comfortable. Now, if you have any doubt in your preparation strategy, so you may ask to me.
Yes,
And
sir.
if you are comfortable in Hindi, then we can talk in Hindi also. Yeah.
हाँ सर, हिंदी समझता हूँ सर।
अच्छा,
थोड़ा
समझते हैं
थोड़ा थोड़ा
हिंदी?
बात कर। हाँ हाँ सर,
ओके।
लेकिन
सो
बोलने में थोड़ा ग्रामेटिकल एरर्स
सो
आ जाते हैं।
इट्स ओके, इट्स फाइन, इट्स फाइन। हाँ, चलेगा।
ओके सर। ओके सर।
प्रेम, पहले तो मैं ये जानना चाह रहा हूँ कि कैसा लगा अभी आपको मॉक इंटरव्यू दे करके?
Sir, uh, the first thing is I'm getting that sense of relief, sir, because ये मेरा पहला इंटरव्यू है सर। मैं कभी जॉब इंटरव्यू भी नहीं दिया है सर। ये मेरा
ओके।
पहला पहला इंटरव्यू है सर। So I don't know how to face a panel itself, and this happened as a practice session for me, and it's quite relief for me. So
ओके।
अब मैं थोड़ा सा कॉन्फिडेंस से मैं जाकर पैनल को फेस कर सकता हूँ सर। And second thing, sir, the questions that you came out, it has reminded me that where my preparation is lacking and what all the aspects that I have to focus upon. Because I've just, sir, as I mentioned, plant biotic was my area of interest, but there is no such thing called as specialization in our MSc biotechnology, sir. We were supposed to study all the subjects like biochemistry, micro, molecular, everything. So there is no such thing. Just because I felt a little ease in understanding concepts and thing in plant biotechnology, I have mentioned that. And so the second thing is that uh, like thank you so much for being flexible and assigning flexible hours for me, because I know I was troubling you from past 2 3 days, like changing my schedule. So I hope you don't mind, sir.
हाँ,
Yeah.
इट्स फाइन, इट्स फाइन, इट्स ओके। प्रेम, आपके इंटरव्यू के बाद से जो मुझे सजेशंस आपको देना है, व्हाट यू आई वुड लाइक टू suggest you the thing that is, suppose you are telling to your interviewer that you are good enough in plant biotechnology, right? So
Yes, sir.
suppose the statement like that, that means the interviewer will expect to some questions from this topic itself, and you have to answer
Oh,
that. So
okay, sir.
अभी आपने आपकी जो बाकी वैसे जनरल अवेयरनेस की जो कॉन्सेप्ट है वो काफ़ी अच्छी है, एज यू आर एस्पिरेंट्स ऑफ यूपीएससी एंड स्टेट गवर्नमेंट एग्जामिनेशन। सो आपने
Yes, sir.
कर्नाटका के बारे में अच्छे से बताया, आपने क्रिकेट के बारे में अच्छे बताया, आपने अपनी हॉबीज एंड अपने ड्रॉबैक्स के बारे में अच्छा बताया।
Yes, sir.
लिटिल बिट थोड़ी सी ज़रूरत है आपको प्लांट बायोटेक्नोलॉजी के जो कॉन्सेप्ट है उनको ब्रशअप करने की, राइट?
Okay, sir.
अगर आप यहाँ पे
Okay.
सपोज इंटरव्यूअर विल आस्क फॉर क्वेश्चंस लाइक व्हाट इज योर एरिया ऑफ डोमेन और एरिया ऑफ इंटरेस्ट एंड फेवरेट टॉपिक। सो अगर आपसे ऐसा क्वेश्चन ऑफर किया जाता है तो आप अगर इस तरीके से बोलते हैं कि सर हमारा प्लांट बायोटेक्नोलॉजी या डीटी टेक्नोलॉजी या बायो प्रोसेस इंजीनियरिंग अगर आपका इंटरेस्टेड है, तो डेफिनेटली उसके जो बेसिक टर्मिनोलॉजी है ना उनको आपको पता होना चाहिए, राइट?
Okay,
और
sir.
अगर आपको लगता है कि आप कंफर्टेबल हैं बायोकेमिस्ट्री में, इम्यूनोलॉजी में, मॉलिक्यूलर बायोलॉजी में, तो आप वही बोल सकते हैं, राइट? या आप
Okay, sir.
इस तरीके से ही बोल सकते हैं कि सर देर इज नो एनी स्पेशल सब्जेक्ट और डोमेन, बट आई फील लिटिल बिट comfort in plant biotechnology,
Okay, sir.
right? To use such kind of a
Okay.
statement, sir, I am not any expert of any subject, and I am not interested in any more subject like, but I have a little bit interest in plant biotechnology. सो
Okay, sir.
प्लांट बायोटेक्नोलॉजी में आपको क्या क्या करना है? सबसे फर्स्ट ऑफ ऑल तो प्लांट बायोटेक्नोलॉजी में आपको जो जो चीज़ें पढ़नी है, लाइक आपके सिलेबस में
Sir,
बहुत ज़्यादा तो नहीं दिया
one
हुआ
second,
है।
sir. Give me a second, sir. 1 second, sir.
सो प्लांट बायोटेक्नोलॉजी में जो चीज़ें आपको सबसे पहले इंक्लूड करनी है वो ये है प्लांट टिश्यू कल्चर के जो बेसिक स्टेप्स हैं। ये वीडियोस मैं आपको
Okay,
दे
sir.
दूंगा, बट आप उसे नोट करना चाहें तो कर लीजिए। प्लांट
Yes,
टिश्यू
sir.
कल्चर्स के लिए, और जैसे प्लांट टिश्यू कल्चर में कई टाइप्स आते हैं, जैसे एंथर कल्चर है, कैलस कल्चर है,
Yes, sir.
राइट,
Yes, sir.
आर्टिफिशियल
Yes,
सीड
sir.
है, राइट। ये सारे जो
Yes, sir.
टर्म्स है
Yes,
ना, ये सारे टर्म्स आपको
yes, sir.
बहुत अच्छे से पता होना चाहिए कि सीड, आर्टिफिशियल सीड क्या होता है, एंथर कल्चर क्या
Yes, sir.
होते हैं, कब यूज करते हैं, कैलस कल्चर क्या
Yes, sir.
होता है, कब इसको आप यूज किया जाता है। ओके,
Yes,
सो
sir.
ऑल द डिफरेंट काइंड्स ऑफ different tissue culture used in plant biotechnology, you have to must mention it.
Yes,
सेकेंडली,
sir.
जेनेटिक इंजीनियरिंग में आप डिफरेंट काइंड्स के प्लांट बेस्ड वैक्टर यूज करते हैं। उन वैक्टर्स का आपके पास
Yes,
एक
sir.
लिस्ट होनी चाहिए, लाइक एज टी
Yes,
आई
sir.
प्लाइमेट आपने बताया, आर आई प्लाइमेट आपने बताया, तो उनके बारे में ड्रॉबैक्स, उनकी लिमिटेशन की, कितना कोई कैपेसिटी होल्ड कर सकता है, क्या उनकी ओरिजिन है। जैसा कि आपको मालूम होगा कि टी आई प्लाइमेट वन ऑफ द कॉमन एंड फेमस प्लांट बेस्ड वैक्टर विच आर यूज इन द
Okay, sir.
जेनेटिक इंजीनियरिंग, राइट। सो इसको
Okay,
आप
sir.
नेचुरल जेनेटिक इंजीनियरिंग के नाम से भी जानते हैं। ऐसा क्यों बोलते हैं? जो क्राउन गॉल डिजीज हो रही है उसके पीछे का जो मैकेनिज्म है मॉलिक्यूलर, व्हाट इज द बिहाइंड मैकेनिज्म, आपको ये भी जानना हो, यू मस्ट नो अबाउट दिस, राइट। आपको पता होना चाहिए कि हाँ, कैसे एसिटोसाइनिन जो कंपाउंड है वो आकर के उसको एक्टिवेट कर रहा है, वीनस जीन एक्टिवेट हो रहा है, फिर कैसे टीआई प्लाज्मिड का रिमूवल हो रहा है, फिर जाकर के प्लांट की बॉडी में इंटीग्रेट हो रहा है। सो ऑल दोस थिंग्स
Okay,
आपको
sir.
अच्छे तरीके से पता होना चाहिए। तो
Okay, sir.
आपको तो पहले टिश्यू कल्चर के जो टाइप्स हैं उनको आप फोकस कर लीजिए, वेक्टर जो
Yes,
हैं
sir.
उनको आप फोकस कर लीजिए, उनके उनके बारे में अच्छे तरीके से। और
Yes, sir.
जेनेटिकली मॉडिफाइड जो फूड हैं, जेनेटिकली मॉडिफाइड जो क्रॉप हैं, उनके बारे में आइडिया आप ले लीजिए। आपने लिमिटेशंस
Yes, sir.
अच्छा बताया, उसमें कोई डाउट नहीं था। लिमिटेशंस आपने बताया कि हाँ पॉजिटिव और नेगेटिव चीज़ें क्या हैं। इतना सारा
Yes, sir.
चीज़ अगर आप करके जाते हैं, और साथ साथ में जैसे आपकी जो ड्रॉबैक्स हैं, आपने बताया कि आपके साथ में आप एक इंट्रोवर्ड हैं, आप जल्दी किसी से खुल पिल नहीं पाते, राइट। और सब्जेक्ट में कौन सा सब्जेक्ट वैसा है जो कि आपको थोड़ा परेशान करता है, जिसमें आपका इंटरेस्ट नहीं है, उसको बारे में भी आप मेंशन कर लीजिए, हो सकता है आपसे सवाल ये पूछा जाए।
Okay, sir.
अगर आप इतनी चीज़ों पे वर्क कर लेंगे तो डेफिनेटली आपका बाकी चीज़ अच्छा होगा। कम्युनिकेशन में कोई प्रॉब्लम आपको नहीं है, आप अच्छे से बोल पा रहे हैं। और
Yes, sir.
एज यू स्टेटेड दैट इट वाज योर फर्स्ट इंटरव्यू, बट काफ़ी कॉन्फिडेंट दिखे आप। ऐसा कहीं मुझे नहीं लगा कि जहाँ पे यार आपको लैक ऑफ कॉन्फिडेंस हो रहा हो। तो इंटरव्यू वाज
Okay,
गुड।
sir.
और ऐसा ऑब्वियसली होना ज़रूरी है, और होता है सबके साथ में कि 2 4 6 क्वेश्चंस का आंसर आप नहीं दे पाएंगे, राइट। तो
Okay,
उसमें
sir.
आपको बहुत ज़्यादा नेगेटिव होने की जरूरत नहीं है। हाँ, जिन जिन चीज़ों को मैंने आपको सजेस्ट किया है आप उनको फोकस कर सकते हैं। टाइम अगर आपके पास है, कब है इंटरव्यू आपका? 15 को?
16, sir.
16 को आपके पास टाइम है अभी। आप उसको अच्छे से
Yes.
रिवीजन कर लीजिए, ठीक है। डेटा अगर
Yes,
नहीं
sir.
हो तो गूगल से भी सर्च कर सकते हैं प्लांट वेक्टर की लिस्ट को लेकर के, और टिश्यू कल्चर के जो टाइप्स हैं, कोई बेसिक्स वाली जो बुक होंगी उनको ही पढ़ लीजिए आप, ठीक है। और
Yes,
प्लांट
sir.
बायोटेक्नोलॉजी में सबसे बड़ा जो जो कंट्रीब्यूशन है वो कौन से साइंटिस्ट का रहा है, क्या उन्होंने किया है, ये सब थोड़ा सा आपको आइडिया होना चाहिए,
Yes, sir. Okay, sir.
ठीक है।
And yes, sir.
या बताइए।
Uh, sir, is it okay to mention that I was an UPSC aspirant, sir?
Yeah, suppose if there is any questions regarding currently what are you doing, so
Yes, sir.
you have to mention that yeah, sir, I am preparing for UPSC examinations and other state government exam. It's fine.
Is it fine, sir?
Suppose you state, yeah, you like as agree that ki you are preparing for a UPSC exam, then they will ask expected to you that they might be possible, they might be asked some questions regarding general awareness, current affairs, etc.
Yes, sir.
And it's good for you.
Okay, sir. Because it was from 2022 to 2023 that's the only year that I have prepared, and even my preparation was not up to the mark because
Okay, okay. So
right from science background I've just jumped into UPSC, sir.
currently you are not preparing for a UPSC exam.
No, sir, from past August I'm totally completely preparing for CGP exam itself, sir. Not
Okay,
UPSC.
okay,
I
okay,
just
okay.
gave an
So,
attempt in
so,
UPSC, sir.
so there is no need to admit this that you are preparing for UPSC exam, because I think you don't have a much idea about the UPSC
Yes, sir.
syllabus or
It
etc.
has been
Right?
just 1 year.
Yeah.
Yes,
So
sir.
leave it. Just sir, preparing for government exam like CGPTM and whatever the concerned
And
biotechnology posters are there. Not
okay, sir.
mentioning
And
UPSC exam. UPSC exam mentioning that means you have an idea about the general awareness, general knowledge, etc.
yes, sir. So I'm just a budding aspirant in UPSC,
Yeah.
so I'm not much aware of all such things, sir. And even my syllabus is just 30-40% completed. I'm not even completed
So
my UPSC
there is
yet.
no need to admit the UPSC aspirants, right?
Okay, okay, sir. And sir, I do have few questions, sir. Um, Sir, if they ask me that even after doing your masters in biotechnology, why didn't you choose your PhD? Why didn't you continue your research? What has to be those convincing reasons to be
That
said
means
by me?
okay. That means ki why you not join the PhD?
Yes, sir.
Yeah, there are so many reasons to not join the PhD because PhD is not a just a course. PhD is more about the patience, more about the time taken, and more about the interest in the research. So there is no compulsion that every plant biotechnology or biotechnology students will go for the PhD. There is no any compulsion, right? So PhD is more about the research, and I don't think sir that my temperament is about that that is research oriented. I am interested in doing some challenging task and doing some government job. And as we know sir, there is huge lacking of a government job after even doing PhD. So
Yes, sir.
PhD is more time taken. Firstly, PhD is that is not about just only degree. It's a dedication and it's a interest because it will take nearly 5 years. So and those second thing that is the person who are going for the PhD will have definitely research temperament and research orientation mindset, right? So I don't think sir that I was keen interested sir. That's why I do not join PhD. सो आपको इस तरीके से बोलना है कि आप नेगेटिव भी नहीं हो और आपका काम भी चल जाए, राइट? जैसे सपोज कि मैंने आपसे बताया कि पीएचडी सिर्फ कोई एक डिग्री नहीं है कि जिसको आपने ज्वाइन किया, एडमिशन लिया, सिलेबस खत्म किया। उसमें पूरी की पूरी तरीके से आपको सारी चीज़ें इंडिपेंडेंटली करनी पड़ती है। देयर इज नीड ऑफ टू बी इंडिपेंडेंट, राइट? देयर विल बी नो एनीवन हु हेल्प यू। सो एंड विल टेक मोर टाइम। सर आई सर मुझे ज़रूरत थी कि मैं जल्दी से अपने आप को कहीं सेलेक्ट करूं गवर्नमेंट जॉब में, और एज माय इकोनॉमिक बैकग्राउंड, सो बेसिकली मुझे ज़रूरत थी सर गवर्नमेंट जॉब की। सो पीएचडी टाइम टेकन लगता, इतना टाइम मेरे पास सफिशिएंट नहीं था। In future, if I will select for somewhere and there will be a chance, then definitely I will join for the PhD as a part time.
And what is the limitations of a private job in biotech sector, sir? Why did you join a private job?
Private
If
job,
not understand.
okay, private job has limitations because in private job like as pharmaceutical, like as dairy industry, like as cosmetic industry, these are the certain area where biotechnology can student can join the private job. In pharmaceutical company, in any kind of private job, there will be a certain, there will be a certain just only work, that is there will be certain fixed work. There is no any use of your knowledge, there is no any use of your intellectual, there is just fixed work. Suppose simply is if I say that that is प्लांट मीन्स बायोटेक्नोलॉजी में प्राइवेट जो जॉब कर रहे हैं वो पढ़े लिखे मजदूर हैं। एक मजदूर होते हैं और एक पढ़े लिखे मजदूर, राइट? आप बायोटेक इंडस्ट्री में जाते हैं तो आपको क्या काम मिलता है? या तो आपको कोई एक फिक्स काम दे दिया जाएगा कि हाँ ये वाली मशीन आपको चलानी है, आप चलाते रहिए, या फिर आपको कोई एक पैकिंग करनी है, आप पैकिंग करते रहिए, या फिर कोई आपको क्वालिटी कंट्रोल करना है तो वो करते रहिए। आपके वहाँ पे एजुकेशन का बहुत ज़्यादा इम्प्लीमेंटेशन नहीं होता है। दूसरी चीज़ ये कि प्राइवेट जॉब में सर रिस्पेक्ट नहीं है। जो ऑनर्स होते हैं प्राइवेट जॉब के, जो मैनेजर होंगे, जो डायरेक्टर होंगे, वो एम्प्लॉई को एम्प्लॉय नहीं समझते हैं, उनके पास कोई रिस्पेक्ट नहीं है। वॉट एवर द डिग्री आई हैव, उनको फ़र्क नहीं पड़ता कि मैंने CSIR क्वालिफाई किया है, मैंने PhD किया है, मैंने MSc किया हुआ है। वो मुझे जस्ट लाइक एम्प्लॉय ट्रीट करेंगे कि मैं एक ऐसा ज़रूरतमंद आदमी हूँ और आकर के यहाँ पे काम कर रहा हूँ। सो डेफिनेटली एंड देयर इज़ नो सिक्योरिटी ऑफ दैट इज जॉब। कभी भी वो टर्मिनेट कर सकते हैं, कभी भी आपको निकाल सकते हैं। हर टाइम पे काम को लेकर के वो आपको बॉसगिरी दिखाते रहते हैं, है ना? कभी अप्रिसिएशन नहीं मिलता है। तो ये सारी जो चीज़ें होती हैं वो बहुत ही मेंटल एक तरीके से डिस्टर्बेंस देती हैं। सो दैट इज़ वाई आई डू नॉट प्रिफर टू गो फॉर द प्राइवेट जॉब। So these are the certain limitations. So there is no respect, there is no freedom, there is no calmness. That is, everything is just going to be ruined.
Okay, sir. And yes, sir. Thank you, sir.
Okay, so Prem, wish you all the best for your interview. If you
Thank
feel
you,
any
sir.
kind of a doubt or any kind of a trouble during before going to your interview, then you can just message me and I will help as well
Okay,
as
sir.
possible. Right?
Thank you so much.
Okay. Hopefully, you're feeling
Thank
right now good, quite better.
Yes, sir. Yes, sir.
Okay. Best of luck for your
Yes, sir.
interview.
Thank you, sir.